Dr. Mazlin binti Rosman. First of all, I will explain about Introduction Practical 1, which is basic concept of statistic and variation. Introduction. Ecology research starts with fundamental methods for collecting environmental data using various measurement tools. Basic statistical techniques are applied to measure tree height, leaf dimension, and size of trees. Detailed analysis of collected data is essential, and statistical representation help simplify the understanding of relationship and correlation among different data sets. Statistics including frequency distribution, tables, mean, variance, standard deviation, and linear correlation are vital for illustrating relationship between variables in ecological studies. For example, leaf vary in size and shape but are usually broad and flat to maximize light exposure for photosynthesis. They are arranged on plants to efficiently capture light and cool the plant with specific adaptation for windy conditions. This study aims to use basic statistical tools and ecology by organizing numerical data in frequency distribution tables and analyzing the relationship between leaf length and width using statistical measures. Next, about objective in this practical. Objective in this practical has three. Number one is to calculate and describe the following descriptive statistics: mean, variance, and standard deviation. Second is to understand the concept of correlation, and last, number three, is to determine the relationship between the two variables. Materials that we use in this practical has a leaf, ruler, weighting balance, and graph paper.
In this practical, there are 3 steps of method that have been followed. First step is 10 leaves were taken, their length and width were measured and recorded. Then the mean, variance, and standard deviation of the length and width of the 10 leaves were calculated. Then the data was tabulated in graph paper. Next step, another 10 leaves were taken and step 1 was repeated. Next, another 3 sets of leaves have done. Then the result was tabulated in data matrix. The last step is the data of all the 30 leaves was used to plot a graph of leaf length versus leaf width. The relationship between length and width of leaf was decided. The type of relationship was investigated, then the result of the relationship has been discussed in the discussion part.
Next is the result. For the first set, the mean of the leaf length is 9.67 with variance of 15.0846 and standard deviation of 3.8839. For leaf width, the mean calculated is 4.57 with variance of 2.7468 and standard deviation of 1.6573. For leaf weight, the mean is 0.404 with variance of 0.0840 and standard deviation of 0.2898. On the second set, the mean for leaf length is 8.79 with variance of 5.3943 and standard deviation of 2.3226. As for the leaf's width, the mean is 4.22 with variance of 1.564 and standard deviation of 1.2506. For the weight, the mean is 0.292 with variance of 0.02048 and standard deviation of 0.1431. Next is the third set. For the leaf length, the mean calculated is 9.5 with variance of 6.6022 and standard deviation of 2.5695. For leaf width, the mean calculated is 3.82 with variance of 1.0951 and standard deviation of 1.0465. For leaf weight, the mean is 0.666 with variance of 0.1124 and standard deviation of 0.3352. This is simple linear regression graph of leaf length versus leaf width. It shows positive correlation for all 3 sets, indicating that as leaf length increase, leaf width also increase.
For discussion, the mean of length and width between different sets of leaves. Based on the differences, set 1 has the lowest mean length value of 0.404 compared to sets 2 and 3, which are closer in value to each other at 9.79 and 9.5 respectively. There is a difference in mean leaf length increase between set 1 and set 2 of approximately 8.386, but sets 2 and 3 have very similar mean length values, indicating that these two sets may have leaves from similar species or conditions, leading to similar growth patterns. The average leaf size in set 1 is much smaller. The leaves in sets 2 and 3 are larger and more similar leaf size. Next, for mean of width, the leaves in set 1 are on average the widest of the three sets, shown by the set's highest mean width of 4.57. Set 2, its mean width is 4.22, which is a little less than set 1 but still rather close. When compared to sets 1 and 2, set 3 presents a bigger difference with its lowest mean width 3.82. Given the close means, it is likely that there is some overlap in the width distributions of the leaves across the three sets. This could indicate similar growth patterns or slight variations in the conditions affecting the leaf width.
What is meant by variance? Variance is a statistical measure that quantifies the spread or dispersion of a set data points. It shows an amount of how the mean of a data set is divided by each individual data points in the dataset. What does the leaf have different measure of width and length means? It is referred to the size variation among the leaf. There is different kind of leaf among the leaf because each one has a particular width and length. Variance is a way to measure this variability, showing how much the width and the length of the leaf differ from their average size. A high variance indicates a wide range of a different size among the leaf, while a low variance means the sizes are more similar to each other. Standard deviation is a measure that shows how much individual data points in a dataset typically differ from the average value. It is calculated as the square root of the variance and it is expressed in the same unit as the original data. A low standard deviation means the data points are close to the average, while a high standard deviation indicates that the data points are spread out over a large range. Importance of having different sets of measurement. It is necessary to have different sets of measurement for the width and the length of leaf because this dimension can vary independently. The width and the length of a leaf are distinct attributes and analyzing them separately allows for a more detailed understanding of the leaf overall size and shape. Different sets of measurement help in accurately assessing and comparing the variability, patterns, and correlation between these two dimensions, leading to more precise conclusions in studies related to plant biology, ecology, and other fields.
To conclude, from this practical, descriptive statistics such as mean variance and standard deviation of length and width of leaf have been calculated using formula. Next, the correlation type of this practical has been investigated, which is the graph show positive correlation. And the last one, the relationship between the length of leaf and the width of leaf was discussed, which is linear relationship.
The biotic factors of an environment are all organisms found within that environment. A group of organisms of the same species found within the same environment is called a population. Population is when different species sharing the same area and interacting with each other and make up a community. One way of studying the interaction of organisms in a community is by taking an inventory of all the species in that area and comparing the size of their population. In this practical, we will explain with a simple concept of quadrat by observing at the abiotic factors of the study site and classify the species of the plant. There are 4 objectives in this practical. First, to introduce the simple concept of quadrat. Second, to enhance the power of observation. Third, to record the difference in plant distribution within the quadrat. Fourth, to infer from natural observation the possible factors that affect distribution and abundance of organisms. And materials that we use in this practical is raffia string, measurement tapes, stick, scissors, and notebook.
In this practical, there is 4 steps. The first one, different types of forest areas at camps regarding were the areas under study. The second one is 1 quadrat of 5 meter multiply 5 meter was made at each area type. Multiple quadrat samples were performed throughout the habitat at several random locations. And the third is all leaves found inside the quadrat were observed and noted. And the last one is the data were used to estimate the population size and population density within the entire habitat.
Next is the result of this practical. This practical have 5 quadrats as its result. The first quadrat is located at the entrance of the forest. From the diagram, it showed that the Cranopteris slenderis have the highest density with 74.65%. The second quadrat is located slightly far from the entrance. Clidemia hirta is the most species found with 56.25% of density. The third quadrant also located at the entrance. The most dense species in this quadrant is Kirtonia fortuni with 58.5%. The fourth quadrant is located a bit deeper into the forest. Clidemia ricta is also the most dense species in this quadrant with 64.52%. The last quadrant, which located a bit deeper into the forest, the quadrant also have Clidemia ricta as the most dense species with 45.45%.
For the discussion, according to Biology Online 2022, quadrant can be defined as a square or rectangular plot of land that has been designated to separate a sample and calculate the percentage of plants and animals that are in the market area. There are two basic requirements of all quadrant techniques. The first one is random placement of the quadrats. The sampling using quadrant method should be done by placing the quadrant randomly to avoid bias and produce a more precise estimation of the population. Second is the standardized size of the quadrant. The size of all quadrats used in the sampling should be the same size to ensure consistency in the data collection. Next, the advantage of using a larger quadrant is large quadrats can include a higher number of individuals and species, thus offering more accurate estimation of the population. Meanwhile, the usage of smaller quadrats can provide more refined data compared to large quadrats, as it allows the researchers to do more precise measurements of species density, coverage, and frequency. However, the disadvantage of using larger or smaller quadrats is it's more prone to errors due to the inappropriate size of the quadrant. Larger species, for instance, need larger plots. Small quadrats may result in too much missed individuals of the species, thus leads to the inaccurate representation of the population.
Next, I will explain about issue concerns ecologists in conducting the sampling methods. First, challenging due to visibility and accessibility, issue uneven distribution, and the need for appropriately scarce sampling plots. Second is sampling method direct like visual counts or indirect like DNA sampling have biases and limitation. Third is extrapolate finding to the entire habitat requires careful assumption while minimizing environmental risk. These two ones remains a crucial ethical concerns. And the last one is different observed may also record inconsistent data. And next, factor that we have cause of distribution. In discussion, the distribution of plant species within a quadrant is shaped by complex interplay of ecological factors operating at various scales, from microhabitat differences to broader environmental and historical influences. Understanding this factor is crucial for interpreting biodiversity patterns and for effective conservation and management of ecosystem. The number of plant species in a small area like a quadrant can vary because different plants prefer different conditions. Factors like soil type, how much sunlight and water they get, and whether animals eat them all affect which plants can survive in each spot. Some areas might have more type of plants because they have a better mix of this condition. Different factors like where the quadrant is located, like how much sun it gets or what the soil is like, whether there have been disturbance like fires or animal eating plants, and how plants compete with each other for space and resources all affect how many types of plants you will find in a small area.
In conclusion, the study using quadrats provided valuable insights into species distribution patterns across different areas at camps. Regarding this finding contributes to our understanding of how biotic and abiotic factors interact to shape ecological communities, highlighting the complexity of natural ecosystems.
My name is Noorana Shuhada, and now I will explain about the competition. Competition refers to something that occurs within two different species or involve members of the same species that use the same limited resource. And competition have three different modes. First, interference. Second, intraspecific. And third, interspecific.
What is the meaning of intraspecific? Intraspecific refers to something that occurs within a single species or involves members of the same species. There are some examples of intraspecific, which are intraspecific competition, second one is intraspecific aggression, and the last one is intraspecific cooperation.
This is the example of intraspecific competition. Our group choose tiger or Panthera tigris. Tigers are rarely compete each other because they usually interact with each other. If this competition happen, tiger tend to compete for same prey, which is their food. This occur among the female tiger to feed their cubs. Next, among the males, they are compete for same mates and territory.
Next, what's the meaning interspecific? Interspecific refers to interaction or relationship between different species. This term is commonly used in ecology and biology to describe various types of interaction, such as interspecific competition, interspecific predation, interspecific mutualism, interspecific parasitism, and the last one is interspecific hybridization.
An example that can be used to describe interspecific competition is tiger versus leopard, where both are considered apex predators in the same region. Tigers and leopards prey on the same animals, thus leading to direct competition for food. These two predators also often share overlapping habitat, thus causing temporal separation to occur between them. The hunting times will differ between these two animals, with leopard hunting time is during night and tiger's hunting time is during dusk or dawn. Being in the same region also led to spatial avoidance, with leopards often avoiding areas occupied by tigers to reduce the risk of direct confrontation.